For coming. My name is Felix Beaudry, and I'm joining you from the University of Rochester. Today, I'll talk to you about some of my work on sex chromosomes from my PhD, which I completed at the University of Toronto. I'd like to begin with a language note. Sex chromosomes often carry with them significant social meaning, so I'd like to make clear that a person's gender is separate from their sex, and it's not determined by sex chromosomes. A person's phenotypic sex is also not determined by sex chromosomes alone, but by an interplay with hormones and anatomy. Since I'm not talking about humans today, I'll make the simplifying assumption that sex chromosome set correlates with sex phenotype, but it's important to remember that this is not always the case. I think the reason sex chromosomes have captured the public imagination is because they stand out from all other chromosome pairs. Here I'm showing you the chromosomes of three very distantly related organisms. We've got an insect, a bird, and a plant. And we can always identify the sex chromosomes. Sex chromosomes are the only heteromorphic pair. My research investigates this pattern. What is it about sex that causes the sex chromosomes to pop out to anyone looking under a microscope? One of the key players is recombination. The X and the Y are the only chromosomes with restricted recombination over the majority of their length. The absence of recombination allows the X and the Y to diverge, leading to their very different shapes. One prominent hypothesis to explain the absence of recombination between X and Y is that some alleles are better when they're inherited by one sex more often than by the other sex. If alleles are more beneficial to males than to females, those alleles will do better if they are linked with maleness, and the Y will benefit from lowered recombination with the X. This is actually consistent with what we see in some plants. Some of our previous work has shown that the Y chromosome of the plant Rumex stachylus is enriched for genes with pollen bias expression compared to the rest of the genome. Pollen is a key male reproductive structure in plants and may therefore benefit from being inherited with maleness. But this observation still leaves us with some questions. How does lowered recombination between X and Y evolve? The classic model would suggest that the benefit of inheriting pollen beneficial alleles with maleness is enough to favor the spread of a genetic element that restricts recombination. This would lead to the canonical recombination suppression between X and Y. However, an alternate hypothesis that hasn't classically been given much attention but is starting to gain some traction proposes that sex chromosomes are more likely to establish in regions that already have low rates of recombination. This hypothesis doesn't require the spread of elements that restrict recombination. New male beneficial genes in these regions of low recombination are immediately more likely to be linked to maleness. Working in the lab with Stephen Wright, Joanna Rifkin, and I set out to test these hypotheses in the plant Rumex stachylus. Rumex stachylus is perfect for this kind of inquiry because it's made up of two subspecies with different sex chromosome sets. While males from the western range have an X and a Y, males from the eastern range have a second Y chromosome. The second Y arose after an autosome fused with the X, leaving its homologue to segregate as the second Y. As a quick note, I'll be using green and blue colors to connote the XY and XYY subspecies throughout the rest of the talk. This unique feature of the sex chromosomes of Rumex stachylus allows for two key comparisons. We can compare both ancestral X chromosomes to each other to get a sense of what the autosome that became the X might have looked like before it was the sex chromosome. And two, we can compare the neo-X chromosome to the homologous autosome, which is not sex-linked, to get a sense of whether the recombination landscape changed with sex linkage. To look at recombination in our two subspecies, we take a three-pronged approach. We first complete the first chromosome scale assembly of Rumex stachylus. This assembly was made using a male from the XY subspecies, so based on chromosome counts, we can expect 4 autosomes and a smaller X. Next, we performed separate crosses and sequenced over 100 offspring in each subspecies to create linkage maps for both of our subspecies. This is a direct measure of recombination, but can only measure a limited number of recombination events. To confirm whether the patterns of recombination extend at the population scale, we also infer recombination from linkage disequilibrium using genetic polymorphism data for over 200 individuals from across the range. As our first step, let's look at our genome assembly and identify the sex chromosomes in the XY subspecies. As expected, we recovered 5 chromosomes in our assembly. To locate the sex chromosomes, we perform both quantitative trait loci analysis using our cross and genome-wide association analysis using our polymorphism dataset. These independent datasets identify the same chromosome as significantly associated with sex phenotype. This fourth panel here with green vertical bars, which I've labeled X. Now let's add our recombination data. The bottom 2 panels are the GWA and QTL from the previous slide. The middle panel shows centimorgan distance per megabase estimated from our linkage map. And the top panel shows our estimate of population-scaled recombination rate estimated from linkage disequilibrium. In both panels, low values represent low recombination. We can see that both datasets suggest low recombination on the sex-linked part of the X. These are sex average recombination estimates, so the results cannot be explained by low rates of recombination between X and Y, suggesting the sex-linked part of the X chromosome shows low rates of recombination in our XY subspecies. Now let's add data for our other subspecies, the one with the neo-X. Here in blue, we want to identify the 2 chromosomes based on our XY subspecies assembly that correspond to the single large fused X in the XYY subspecies. As expected, we see that the chromosome we identified as the X chromosomes in our XY subspecies shows significant association with sex in both the QTL and GWAS in our XYY subspecies. Also, one chromosome that we identified as autosomal in the XY subspecies now shows significant association with sex in both the GWAS and the QTL in the XYY subspecies. This is this chromosome to the far right. This is the fused neo-X part of the X in the XYY subspecies. Now let's zoom in only on these 2 chromosomes to look at our recombination. Again, the bottom 4 panels show sex association, so that's our genome-wide association study, and our QTL. The middle panel shows the genetic distance by megabase based on our cross, and the top panel shows our population-scaled estimate of recombination. What we see is that on the ancestral X, both subspecies, the green and the blue, show low recombination in the part with significant sex association. Let's add our neo-X to the right. We see that the region that is consistently associated with sex has low recombination on the neo-X chromosome, but also, and this is what we were most curious about, we see low recombination on the homologous autosome in green. This result suggests that even without any sex linkage, we see low recombination on this chromosome, or on this part of the chromosome. The autosome that was to become the X likely already had low rates of recombination. Overall, we looked at both an ancestral and neo-X chromosome and found evidence suggesting low recombination on both. Low recombination preceded sex linkage on our neo-sex chromosome. Our results are in line with the alternate hypothesis. Low recombination precedes sex linkage. Preliminary work in papaya and kiwi find a similar mechanism may be at play, suggesting this could be an important path in sex chromosome evolution in plants, but this will take more investigating. I'd like to thank my collaborators on this project, Joanna Rifkin, Stephen Wright, Spencer Barrett, Zoe Humphreys, and Baharul Chowdhury, as well as the Wright Lab at the University of Toronto. And with that, I'd love to take any questions.